Proteins are essential to the structure, function, and regulation of the body's cells, tissues, and organs. They are used in numerous of ways in a living organism's body or structure. Protein functions include growth and maintenance of tissues such as bones and muscles, the control of biochemical reactions such as cellular respiration controlled by enzymes, delivering signals from brain to other body regions in the form of hormones, providing rigidity and stiffness to cells and tissues, regulating concentration of acids and bases in the blood and other bodily fluids, helping form antibodies to fight infection, serving as aiders in active transport to move large molecules across a semi-permeable membrane, controlling different characteristics such as hair type and colour, eye colour, and it's used in nails, scales, and so forth. These essential proteins are made in a process called polypeptide synthesis. The name polypeptide refers to numerous amino acids in a chain which form a protein, plus synthesis, meaning production. So essentially, polypeptide synthesis is the production of protein produced to determine the physical, physiological, and behavioral traits of an organism. Polypeptide synthesis involves two key stages. Transcription, which is the process by which genetic information stored within a double helical DNA becomes transcribed into a single-stranded mRNA within the nucleus. In simple language, transcribing is a conversion of something without altering the meaning, such as converting a speech to handwriting. The genetic information is stored as complementary nitrogenous base sequences within the DNA nucleotide composition. The nucleotide monomer in DNA consists of a backbone made up of deoxyribose sugar, a phosphate group, and a nitrogenous base. Due to there being 4 different bases, there are 4 different nucleotides. The bases are complementary in the fact that adenine joins with thymine and cytosine joins with guanine. These base sequences code for 1 of 20 amino acids, 9 of which need to be taken in through our diet as the body can't manufacture them. DNA, however, is fixed within the nucleus and cannot move. This is why the information needs to be transcribed into an mRNA strand, whereby it can then exit the cell and be used during the next phase. Translation, which is the process by which the code carried by the mRNA becomes translated into a specific amino acid. Both of these phases occur in 3 stages: initiation, elongation, and termination. Let's look at the whole process a little closer. Within the eukaryotic cell, there is an organelle called the nucleus, which stores a complete set of DNA. This is called a genome. Within this nucleus, chromosome pairs are stored. Chromosomes are long coiled up strands of DNA tightly coiled around proteins known as histones that give it the structure. The tight packaging allows for 2 meters of DNA to be stored within such tiny cells. DNA is stored as a spiral double helix, once again to save space. Not all DNA is used in polypeptide synthesis. Only about 2% of it is used to code for protein. Specific sections of DNA called genes are what code for protein. Genes carry the inherited protein building instructions. An enzyme called RNA polymerase transcribes the gene. Particular sequences of nucleotides in DNA forms a code. Codes are triplets of nucleotides. This order determines the amino acid which is sequenced, and this sequence determines which protein is formed. Promoter regions or codes on the gene inform the enzyme where to start transcription. This is almost always the amino acid methionine, coded by AUG, and terminator regions or codes inform the enzyme where to stop transcribing the gene. The first stage in transcription is initiation. RNA polymerase joins to the start of the gene when it becomes activated. The gene becomes active by being turned on through a process called gene expression, whilst other genes become deactivated by being silenced through a process called gene regulation. RNA polymerase unwinds the double helix, and another enzyme called RNA helicase works in conjunction with RNA polymerase to unzip the strands by breaking the weak hydrogen bonds between the bases, causing the 2 strands to separate and expose the nucleotide bases for transcription. The template strand is read 3' to 5', and the coding strand is read 5' to 3'. Elongation. RNA polymerase moves along the 3' to 5' template strand and adds free-floating new nucleotides found in the nuclear sap complementary to that of the template RNA strand and promotes attachment and elongation. The coding strand has the same nucleotide base sequence as the mRNA, apart from thymine being replaced by uracil, as the mRNA molecule consists of uracil instead of thymine, like that of DNA. Only the template strand will be transcribed, as the mRNA will be mirroring the coding strand, which contains the sequence for the amino acids to be built, which is being replicated or transcribed, so it can leave the nucleus and enter the ribosome where the protein shall be built. The two strands of DNA rewinds and recoils behind the RNA polymerase as it moves downstream the DNA. Termination. The RNA polymerase encounters the terminator region or code, which signals the end of transcription. This newly synthesized mRNA undergoes post-transcriptional modifications. Before translation, the non-coding or template strand needs to be edited or spliced. Non-coding sections called introns are removed and the remaining exons become spliced together to form mature mRNA. This is the strand which will serve as the template for replicating the code. The mRNA strand is capped by adding a guanine nucleotide at the 5' end of the primary mRNA strand to protect it from degradation of enzymes during transport. A poly-A tail, which consists of multiple adenosine nucleotides, is added to the 3' end, which slows down degradation by cytoplasmic enzymes. The freshly synthesized mRNA strand exits the nucleus through a nuclear pore and enters the cytoplasm until it reaches a ribosome and binds itself to it for the next phase. Translation. Ribosomes are made up of rRNA or ribosomal RNA and proteins. It is these structures which function to synthesize proteins. Ribosomes can be found floating within the cytoplasm or attached to the endoplasmic reticulum. Ribosomes are composed of two subunits called the small subunit and the large subunit. Initiation. The mRNA is scanned or read by the ribosome, which associates both subunits of the ribosome to service the mRNA molecule. The starting or promoter codon AUG is identified on the mRNA, and a tRNA molecule is brought to the complementary anticodon UAC, carrying the relative amino acid methionine. tRNA or transfer RNA molecules are found within the cytoplasm and carry the complementary anticodon to perfectly match each codon on the mRNA, allowing them to join. On the end of the tRNA is an amino acid attachment site, which is where the specific amino acid is bound to. Each mRNA codon specifies an amino acid. It is easy to determine which amino acid is being coded for by using an mRNA codon chart. This is used by reading the first, second, and third base of the codon. Elongation. After the first amino acid has been added, a second tRNA comes in and a peptide bond joins the previous amino acid to the new one into a chain. The first tRNA molecule dispatches from the amino acid and leaves to find another amino acid for translation, either in the same ribosome or in another ribosome, where a different mRNA strand is being synthesized. The ribosome moves the mRNA along and a third tRNA attaches to the codon. Another peptide bond links this amino acid onto the chain. The second tRNA molecule dispatches itself and leaves behind its amino acid. As the peptide chain elongates, the ribosome is divided into 3 sites. The A site is where the new tRNA molecules enter the ribosome. The P site is where peptide bonds are formed. The E site is the exit site from which empty tRNA molecules, whose amino acids have already bonded with existing amino acids, exit the ribosome. This process continues as each new amino acid forms bonds with the previous one, forming a polypeptide chain. Termination. One of 3 stop or terminator codons, either UAA, UGA, or UAG, is detected, triggering several events such as the last tRNA dispatching from the mRNA strand. This is the completed polypeptide chain. The ribosome dissociates when releasing the polypeptide chain. The messenger RNA may enter another ribosome and repeat the protein synthesis process, or it may be broken down and its subunits may be reused. Protein structure. After the polypeptide chain has been released, it undergoes certain structural modifications to form a functional protein. The sequence of amino acids determines the 3D shape of the protein, as a specific amino acid sequence spontaneously folds the protein into its 3D shape. There are 4 levels to a protein structure. Primary structure, which includes the amino acid sequence. Secondary structure, which includes the arrangement of amino acids into structures such as alpha helices or pleated beta helices. Tertiary structures, which include the major folds and bends within the polypeptide chain. Quaternary structure, which is the combination of several polypeptide chains to form a protein molecule. The importance of polypeptide synthesis. Polypeptide synthesis is vital to functioning organisms as proteins enable the correct structure and function of an organism. A change in DNA sequence is where one or more nucleotides are deleted, inserted, or substituted for different nucleotides. This results in a change in the sequence of the nucleotide bases, which alters the order that the amino acids are coded for, which will change the shape of the protein. This means the protein won't function properly or won't function at all. Proteins of incorrect structure and function result in an organism not developing or surviving. Protein shape is important to their function. Therefore, the right amino acid sequence is important as well as the right mRNA codon sequence, and hence DNA is important. Any change in the structure or the amount of DNA of an organism is called a mutation. Mutations are caused by mutagenic agents like chemicals, ultraviolet light, etc. Cancer is an example where these mutagenic agents have affected this molecule, allowing for rapid replication, aiding in the spreading and harmfulness of cancer. Mutations can either be harmful due to stopping or altering the production of proteins. An example of this is albinism, where it leaves organisms with no pigmentation. This could make this organism more susceptible to prey and hence discontinue the species' life cycle. Mutations can also be beneficial by helping the survival of an organism, such as viruses. Their constant changing of coding gives rise to new strains which avoids detection by the host's immune system.